Hello everybody, welcome to NEET Simplified. So today I am here again with another episode to simplify another confusing concept. So here, today we are going to talk about the Malvaceae plant family, and we are going to discuss the most confusing part over there. We are going to discuss about the floral parts, which are always a little confusing. Let's simplify that in this episode, and by the end of this, you're going to be clearly understanding everything in the Malvaceae inflorescence. So here, let me very quickly tell you why the name Malvaceae. You see here a flower, isn't it? We call this flower by the name mallow. This is the mallow flower, and Malvaceae gets its name from this flower. And whenever we think about Malvaceae, we always get reminded of only one beautiful flower, the hibiscus, isn't it? So we generally call this as the hibiscus family as well. And what about the inflorescence here? We call this inflorescence as solitary cymose. The inflorescence is called solitary cymose, and today we will be talking about in detail about the parts of the flower here, which are always confusing. So let's very quickly dive into this topic and simplify that. So, we are going to begin with the same order always, as we begin with the Calyx, Corolla, Androecium and Gynoecium. But before we start with the Calyx, there's very important thing which we have to discuss about here. Firstly, the flowers are bractate here. So let's understand about the presence of bracts. As you can see here, we have a pedicel, isn't it? We all know that this stalk is called as the pedicel. At the base of the pedicel, you are finding the bracts. So the flowers are definitely bracteate, right? So you're finding the bracts. Now what about these appendages which you are seeing here? These are not bracts. Mind you, we have to remember that there are certain short appendages around the calyx. So here you are seeing the calyx, and surrounding the calyx we are going to find these structures which we call as bracteoles. We are going to find a circle of bracteoles surrounding the calyx. And what is its role? It is protective in nature. It is going to protect the bud. Yes, now we are going to see that this whorl of bracteoles is going to be called as epicalyx, which is surrounding the calyx. Right? Now coming to the calyx, we are going to find 1, 2, 3, 4 and 5. We are going to find 5 sepals. Yes, when you cut open the calyx, you're going to find this picture. You're going to have 5 sepals there. And also, you will see that all of these are not individual, isn't it? They are all fused. They are all fused, making a tube. So, we will call this condition as gamosepalous. The sepals are fused, hence we call this as gamosepalous. And fusing what? Fusing and forming a tube. Right. Now, this tube is going to protect the internal organs. Like you can see here, a mature bud which is being protected by the calyx and also the epicalyx. Yes, now that's about the calyx. Also, let me tell you the arrangement of this calyx over here. We call this as a valvate estivation. We have the arrangement which is called as valvate estivation for the sepals. Now, this is something which you have to remember because there's another kind of similar thing which we will be again facing about it, or again we are going to read about something called as estivation again. So remember, sepals are arranged in a valvate estivation. Now moving to the most colorful part, the corolla. So here we are with the corolla, the beautiful part of the flower. Yes, here in hibiscus you are seeing 5 petals. Right, you're seeing 5 petals. First point. Secondly, they're all free, isn't it? They are all free, not fused. So we call this as polypetalous condition. Now coming to the arrangement, this has got a specific arrangement where you're going to see one edge of the petal is facing inwards, the other edge is facing outwards. For each and every petal, you are seeing the same thing. You're going to find one edge facing inwards and the other edge facing outwards, and this particular arrangement, we call this as twisted estivation. Right? So this is called as twisted estivation. The petals are free. Now there is a small twist over here. We are seeing the petals are definitely free. When you look at the flower, you will see that the petals are free. They're not fused. But once you go down to the bottom of the petals, at the base, you will see that the petals, the base of the petals are fused to something called as a staminal tube, right? And this condition is called as adnate condition, as it is fixing at the base of the petals. We call this as basally adnate. Now when you look at it from the top view, you're not going to understand it. So we will take a longitudinal section. Yes, so here you have the longitudinal section. Now what you're finding here, the red part is the base of petal. Here's another petal. We are taking a longitudinal section of the flower. You're seeing two petals only. So here you have the base of the petal. Now this base is fusing to a staminal tube. So you have a staminal tube here. Now this is what we call as the adnate condition. You were seeing that this base of the petals is fused to the staminal tube. Now what is all this staminal tube and what is happening here? We're going to understand it clearly in another slide. Yes, when we hear the word staminal tube, it is related to the stamens. But before going there, let us see how are these stamens structured. Now you're going to see the stamens with the anthers that are reniform. And what is reniform? Which is nothing but these are kidney shaped anthers, which you can see here very clearly. And you have got the filament which is fixed at the back side. That is, it is dorsifixed filament, right? Now what about the number of stamens? You will see multiple stamens, and we call this condition as infinite condition, where you have got like innumerable stamens. We cannot really count them, so we call that as infinite condition. Now coming to the anthers, you can clearly see in this image about the kidney shape, and also you will see that these are monothecous and monolobed. Until now we discussed about dithecous and bilobed anthers, right, which are going to have something like this. They are going to have 4 sporangia in them. If at all, it is bilobed and dithecous. You are going to find a tetrasporangiate condition here. But in case of the Malvaceae, you are going to find bisporangiate condition. There are only 2 sporangia here, right? So as you can clearly see in this picture, there is only one theca, monothecous. There is only one lobe, monolobe. And you're seeing only 2 sporangia from which the pollen is releasing out. In which way? In a transverse dehiscence manner. You're going to see the dehiscence is happening in a transverse manner. Till now we have seen longitudinal dehiscence. Here you're going to see transverse dehiscence with monothecous, monolobed anther, which are going to have 2 sporangia only. Right, if at all. So please take a note. Remember, if at all it was bilobed and dithecous, you will find a condition this way. right? But here, as we have only two sporangia, we are going to find it is to be monothecous and monolobed. We are going to see the pollen releasing from the bisporangiate anthers in this way. Yes, now coming to the tube which we have discussed earlier. So we have finished about the anthers. Now, what are these filaments doing in the stamens? You will see that the filaments which you can see here, the filaments which are all fusing, as you can see here as well, there are multiple stamens and all the filaments you are seeing that they are all fusing to form a single bundle. You will see that there is a single bundle which is formed by the fusion of these filaments. This condition is called as monodelphous. And what are they fusing to form? They are fusing to form a staminal tube. As you can see here also very clearly, you are seeing the multiple stamens, their filaments fusing to form this long staminal tube. Now, as we have understood previously, we have discussed about something called as the basally adnate condition of the petals, and we have seen even the staminal tube in the longitudinal section. We have seen the basally adnate condition of the petals. So these are the petal bases. And this is our staminal tube. Right. And also one more special feature for the staminal tube is that you will see the style of the ovary. As you can see this here. So this is our gynoecium you are seeing here. The style which you can see here as well. It is passing through the staminal tube. Right. So the basally adnate condition has to do something with the staminal tube, isn't it? Also, the second feature of this is that the style of the ovary passes through the staminal tube. Like how your ballpoint pen works. You have your pen and the refill. They are not fixed or attached anywhere, but the refill is going to pass through your pen and both work together. Right? In the same way, the style of the ovary is going to pass through the staminal tube just like the refill. Now coming to the gynoecium. This is going to be a very special discussion, so you have to pay a little more attention. Yes? So in the Malvaceae family, we are going to find a single ovary. As you can see here, we have a single ovary, right? We have the style passing through the staminal tube also. That also we have discussed earlier. Now, as you can see here, there is a central axis in this ovary, isn't it? And you're finding multiple ovules which are all attached to this central axis. So, we will call this placentation as axile placentation. Yes, so that's about the ovary with multiple ovules all placed on this axis. But now when you look at this way, we are not able to understand it clearly. So what we will do, we will take a transverse section. We are going to look at the top view of it. We are going to take a transverse section. So this is from the microscope. Let's understand it very clearly. When you look at it from the top, we are finding 1, 2, 3, 4 and 5. So we are finding 5 carpels, right? So hibiscus has got 5 carpels, and we are finding the chambers here as well, isn't it? So these chambers are also 5, hence 5 locules. You are finding the central axis as well. So this is our central axis from the top. You're going to find the central axis over here. So this is our axis, to which you will see multiple ovules are all arranged onto this axis through axile placentation. Right. Now, This is a little clumsy to understand because everything is so closely attached, right? So for us to get it clear, to understand it clearly, let's go to our kitchen and get our lady's finger. Why? Because it is also belonging to the Malvaceae family. And when you try to cut open it longitudinally, just take a single slice of it, you're going to get this, isn't it? Now, what is all this? Here, we are finding the seat of the floral organs. This is our thalamus, right? Here also in the bhindi you're going to find the same thing. Now what is this? It is going to be a mature ovary. It's nothing but the ovary which is matured. Now that's what we call as the fruit. Now here you are nothing but seeing the same thing like in this hibiscus. So you're going to see here, this is the ovary, right? Sitting on the thalamus, as you can see here. Now, this is our central axis, just like how you have it in the case of the hibiscus. The central axis, you are finding the ovules attached to this axis through what again? Through the axile placentation. Right, again let's take a transverse section of it, which is very easy. We're going to get a slice of it. Sometimes you find like this, isn't it, where there are no seeds attached to it. You can clearly see the carpels here as well, which are 5. So we have 5 carpels, we have got 5 locules as well, and you are seeing the ovules attached. This is our central axis. Now we are taking the top view of it, isn't it, through the transverse section. When you look it from the top, you're going to have the central axis as well. So that's how we have got the axile placentation. Right, now whatever we have discussed so far, just very clearly we are going to revise it, right? But before going for our revision, let me tell you, I hope this is getting clear a bit about the inflorescence, about the flower parts we have discussed. So that probably that is getting cleared so far. If it is getting cleared, don't forget to subscribe to NEET Simplified, and also do like and share this content, because we are the only NEET zone where you're going to find exclusively hand-drawn diagrams and also images directly, which we take through the microscope and present it to you to make your serious and confusing concept to be simple and understandable, right? So drop your all doubts off and don't forget to subscribe and hit the bell icon. Now let's go to the revision. Yes, so we have understood that the thalamus or the receptacle is going to be the seat of the floral organs, as you can see here very clearly. Even in the bhindi we have seen the thalamus as well, right? Now after that we understood that there is a pedicel to which there are bracts attached, isn't it? So we have the flowers are bractate, so we have bracts attached over here. Now, whenever you remember about the bracts, immediately remember about the epicalyx. What is this? It is the whorl of the bracteoles, right, which is protective in nature. So that's about the epicalyx. As you can see, the bracteoles over here surrounding the calyx. Now we understood about the calyx, which is gamosepalous in nature with 5 sepals and also protecting the internal organs. And after that we understood about the 5 petals, isn't it? We understood about the 5 petals, the corolla, the the twisted estivation of it. We have also understood about the adnate condition, the basally adnate condition of the petals. Now we understood about the next thing is the stamens, which are going to be multiple in number. We have seen the anthers, which are all extrose in nature. These are extrosanthus. Why? Because you will see that the dehiscence is happening towards the outside. So they are exposed to the outside. So the anthers are dehiscing also outside. So these are the extrosanthus, and you are seeing all these filaments joining to form the staminal tube. As you can see, the staminal tube, which is very clearly seen here, right? And we understood about one more important condition for the staminal tube, that the style of the ovary is going to pass through the staminal tube without getting fused to it. And here you are seeing the stigma of the ovary. Now very clearly here the anthers and the stigma are in different levels. So you will see that they are promoting cross pollination by hercogamy. Yes, we understood about the single ovary as well with multiple ovules attached onto an axis. We call that as axial placentation. Now, whenever we discuss about the flower, the most important thing to discuss the next is the floral formula. Yes, so let's understand that as well. So as you can see here, we have the bracts. So this is our bract. So you will see the flowers are bractate, and coming to the symmetry, you will see that this is an actinomorphic symmetry. That means the flower can be cut into two equal halves in more than one plane, right? That we will represent in the floral formula. The flower is bisexual containing Androecium and Gynoecium. We are going to mention about the Epicalyx, about the bracteoles, which are in 3 to 8 in number. Then representing calyx by K, there are 5 sepals, gamosepals. The petals 5 in number. Polypetalous, they are free. Androecium infinite in number. So we will write that as they are fusing to form the staminal tube, as you can see here. We will represent that condition. Also the adnate condition, the basally adnate condition of the petals, we are going to represent it here. And coming to the gynoecium, it is a superior ovary, because you will see the rest of the parts of the flower are arising from beneath the ovary. So you will see the ovary is superior in nature, and you will see that the carpels are 2 to infinite. So that is syncarpous in nature. You will see they are all fusing, isn't it? So that is syncarpous in nature. So but that also we are going to represent here in the floral formula. So that's about our floral formula. Now, moving ahead, let's very quickly discuss few tricky questions which are going to be helpful for you. Like if you face such questions in exam, how will you answer this? Let's understand that. So here our first question is, which of the following is a characteristic feature of Malvaceae stems? Mucilage canals, thorns, and we have hollow nodes and glandular trichomes. Now, whenever we remember about Malvaceae, we understood a relatable example at home, the bhindi, isn't it? When you cut it, you will see a slimy layer which is like oozing out of it. Now, what is that slimy layer? It is nothing but mucilage. So, you're going to write the answer would be mucilage canals. Now, whenever there's a tough question, that's why we always emphasize on thinking about relatable examples so that you can answer such difficult questions very easily. Right? So, whenever you remember about Malvaceae, remember the bhindi, you understand about placentation, about the ovary, ovules, and also the mucilage, which is going to be present in the Malvaceae stems. Now, moving to the next question. What is the usual estivation of Malvaceae sepals? So, we understood that there are two kinds of estivation which we have discussed in today's episode. One was for sepals and the other was for petals, right? In case of petals, we understood that they are arranged in a specific manner such that the edge, one edge is outwards and the other edge is inwards. So that was twisted, isn't it? That was for petals. Now coming to the sepals, it is valvate estivation, right? Now moving to the next question. The petals of Malvaceae are typically what? Now we will go through or answer this question by eliminating the options. The definitely D is not going to be the right answer, right? Petals are definitely present. And coming to this option C, they say they are fused. They are free, isn't it? They were polypetalous, isn't it? They were not fused, they were free. They were only fused at the base. There was only adnation at the base, isn't it? So again, A option says that, so this C is wrong. So A option says that adnate to sepal. So this was not the right thing. It is adnate to staminal tube. Hence, your A option also would be definitely wrong. So the right option would be polypetalous. Now, so by this question, we come to the end of this episode. And let me also very quickly tell you, if you want to answer such more tricky questions, go check out our website, which we have given in the description. Please go through that. You will find all the vegetative characters of the plant families and also such tricky questions which can challenge your revision, which can challenge your preparation. Don't forget to like, share, and subscribe. Share this content with your NEET friends. Have a discussion and get all the concepts simplified here at NEET Simplified. We'll meet you again with another confusing concept. We will simplify here like always we do. Thank you. Bye-bye.